Hello, this is a presentation about retinal blood flow imaging with a technology called laser Doppler holography. The content of this presentation is pretty much the content of the two articles in Biomedical Optics Express cited below, so that's where you can find more information. So why is blood flow important? Well, blood flow is what allows to deliver oxygen and nutrients to the body. In practice, the vasculature is organized this way. You have oxygenated blood flow that comes in through the arteries. Arteries then divide into arterioles that divide into capillaries, and the exchange is made at the capillary level. And then deoxygenated blood flow is evacuated by the venous system. Now the whole body needs a very complex vascular system so as to perfuse each organ according to its need that may vary in time. And the problem is that any disruption of blood flow, any decrease of blood flow can be a source of problem because it can provoke a lack of oxygenation. The retina is especially sensitive to a lack of oxygenation because it contains neurons that can die very rapidly. Luckily, it's very easy to image the retina because it's optically accessible, meaning that you can illuminate the retina through the pupil, collect light on a camera, and you can already form some very basic but interesting images of the eye fundus, where blood vessels are directly observable. And the eye is the only part of the body where you can access blood vessels non-invasively, which makes it a very interesting area of research, not only in ophthalmology, but also for cardiovascular application. But just for ophthalmology, we have some reasons that we want to study ocular blood flow because it plays a central role in many critical diseases like age-related macular degeneration, glaucoma, diabetic retinopathy, and the prevalence of these diseases is increasing, and we want to understand better how blood flow plays a role in these diseases because they can lead to a permanent vision loss. So the medical application motivation for measuring ocular blood flow is first to understand what role plays blood flow in the development, and if a decrease of blood flow is the reason of the disease, then it could be used to detect the disease at an early stage before there's any loss of vision, so then a treatment can be given. It could also be used to improve the treatments by finding a treatment that allows blood flow to come to a normal value again, and for that you need to be able to measure blood flow itself. So the ideal ocular blood flow imaging instrument should be able to do the following things. First, it needs to be able to reveal both the retinal and choroidal vasculatures and all of the vessels, which means a high spatial resolution. A second criteria is the ability to quantify blood flow in all vessels in physical units, so either the velocity in centimeters per second or the blood flow itself in microliters per minute. And finally, it would also be very important to measure the pulsatile variations of blood flow, which means having a high temporal resolution. Now, the state of the art in clinical practice, it's first invasive instrument. It's a technology methods that have been there for about 50 years, and they're still used because they have some features that are not met with other instruments. So, for example, to reveal leakages or neovascular proliferation, it's still a very popular method. But because it's invasive, you need to inject something into the bloodstream of a patient, which is very unpleasant, and it's not quantitative, it's not providing measurement with physical units. It's a method that is not so popular actually in clinical. Now, OCT angiography is another technique that is very recent, and this time it's not invasive, so it's able to reveal the microvasculature. It's also angiographic, but it's too slow to study the blood flow fluctuations over cardiac cycles, and it's also not providing quantitative information about blood flow. So you have sort of a black and white map where there is blood flow or there isn't, but it's not quantified. And finally, there is laser spectral flowgraphy, which is technology able to reveal blood flow fluctuations, but the spatial resolution, the flow resolution, and temporal resolution could be improved. So our technology, called laser Doppler holography, is based on laser Doppler concept to make blood flow measurements. And here's how it works. Imagine the eye looks something like this. Inside the blood vessels you have red cells flowing at very high speed compared to the rest of the vessel. And if you illuminate these vessels with a single frequency laser diode, well, the scattered light is going to be affected by the Doppler effect. So the Doppler frequency shift, that value of the scattered wave, depends on the scattering directions, incident and outgoing vectors, and also, more importantly, on the velocity vector itself, so its direction and its value, which means that a greater velocity will mean a greater frequency shift. And in practice, because there's many scattering directions, because there's multiple scattering on the vessel walls and so on, you don't have on a single Doppler frequency shift, but a whole range of frequency shift, which leads to many possible values and a Doppler broadening. And the concept of making blood flow measurements with a laser Doppler is to illuminate the retina with a single frequency laser diode, collect this Doppler broadened beam, and then find some information about the velocity from the statistical properties of the Doppler spectrum. In case of direct backscattering for a velocity of, let's say, 10 centimeters per second, you have a frequency shift of 50 kilohertz, which is kind of a lot, but nothing compared to the laser optical frequency. So how can we measure such a small shift of 50 kilohertz compared to 10 to the 14 hertz is the laser optical frequency? Now, luckily, it's very easy to do with interferences. And that is the concept of laser Doppler flowmetry. With this technology, you illuminate the eye with a single frequency diode, and you make the light that is backscattered self-interfere, because it's considered that this light coming back from the eye is the summation of the contribution of light that has been scattered by red cells, that is Doppler broadened, and light that has been scattered by static diffusers, which do not induce any Doppler frequency shift significant compared to red cells. So this light is coherent, it's able to interfere on the photodiode, and the trick is that the bit frequency of the interfering term is no longer centered on the omega L frequency of the laser, but instead it's centered on the zero frequency, which means that the Doppler spectrum can then be studied. It becomes accessible by the photodiode because it's just a few kilohertz. Now, in laser Doppler holography, the concept is very inspired from laser Doppler flowmetry, but slightly different. Then again, you have this single frequency laser diode, you collect the Doppler broadened beam, but instead of self-interfering, this time you combine it with a coherent reference arm. And these two laser beams are able to interfere on the camera. But this time, because you're introducing a reference arm, you had a lot of coherent gain because the interferent term is proportional to both of the amplitude of this laser beam. So the additional power from the reference arm allows to fill the camera well. So you're not limited by photon noise as you would be if you only had at your disposal the light that is backscattered from the eye, because you don't have a lot of light coming back from the eye. And this time again, because you're making interference, the bit frequency spectrum on the camera is centered again on the zero frequency instead of on the omega L laser optical frequency. Now, to summarize this concept, in laser Doppler flowmetry, the Doppler broadening is measured from the interference between light scattered by red blood cells, which is Doppler shifted, and light scattered by static diffusers, which is non-Doppler shifted. Light self-interfere, but the thing is, the power on the photodiode that you have is only the light backscattered by the eye, and due to laser safety, you cannot send a lot of light, so you don't have much light on the photodiode. You can only make measurements on a single point, and if you want to make a whole image, you need to scan this point across the image, which takes a lot of time, so you have a low temporal resolution. However, in laser Doppler holography, you add an additional reference arm, so you have interference between light scattered by red blood cells, Doppler shifted, an additional reference arm, non-Doppler shifted, and this time it's the configuration of interferometer, and the coherent gain from the reference arm allows to use a camera. So you're able to fill the camera well while making very high frame rate measurements thanks to the reference arm, and this allows to have a very high temporal resolution. So the optical setup of the device looks something like this. It might look complicated at first sight, but compared to the state-of-the-art of ophthalmic instrument, it's very simple. You have a laser diode at 785 nanometer that you split into two using a fiber coupler. On one arm, the object arm, you are going to illuminate the eye, collect the multiply scattered light Doppler broaden. It's combined with the reference arm thanks to a second beam splitter curve on two cameras. And these two cameras, they see exactly the same interference pattern. So on the slow camera, it's used for real time because it's only 80 Hz. It's making a holographic detections, but of very poor quality because the Doppler measurements are completely undersampled. And the sampling frequency of 80 Hz, and we are interested in Doppler frequency shift of tens of kilohertz. It's not making anything interesting, but it's real time. The other camera, on the other hand, it's offline, but it's high quality. So at 785 kilohertz, you're able to sample all of the Doppler frequency shift as should be, but the camera, in just a few seconds, it records hundreds of gigabytes of data, and it's not possible to transfer the data in real time to the computer, so you don't have any visualization, so you have no idea what's going on the camera. That's why we need a second camera for real time. So both of these cameras make holographic detection, and in holography, because you're making interference, you don't just have access to the intensity of the electrical field, but also to the phase. And that allows you to use Maxwell's equations to make numerical propagation, for example. There's other use to that. And we capture the defocus plane of the retina, and we just used the numerical propagation to be in the retinal plane once again, and then we make the laser Doppler measurements that is based on a Fourier analysis. So because Doppler induces frequency shift, it's very natural to use a Fourier transform to analyze the effect on the spectrum. And we do a short-time Fourier transform analysis. Short-time is because we want to resolve the pulsatile changes of Doppler broadening over time. So the statistical properties of Doppler spectrum, they change with the pulsatile variations of blood flow, and that's what we want to measure. So the process is the following. We have our full hologram stack of complex valued holograms, and we slide a short time window along this stack, and for each position in time we calculate the power spectral density from the Fourier transform, and we integrate the highest frequencies of this quantity, and we call that quantity power Doppler. It's the area under the curve. We do this pixel wise. On each pixel we make this analysis, so we then obtain a power Doppler image. It can be interpreted the following way. If there is more blood flow, there is a larger Doppler broadening, so there is more energy at high frequency, so you have a higher value of power Doppler. So with our camera of 75 kilohertz, we are able to run it at 500 by 500 pixels and record about 260,000 holograms on the onboard memory of the camera. So it's a measurement of about 3.5 seconds. And we usually use a short-time sliding window width of about 500 images, which is a temporal resolution of about 7 milliseconds. It can be adapted according to the needs of signal-to-noise ratio. The raw images look something like the images on the left. It's almost only power coming from the reference arm. We do the numerical propagation and we look at image-to-image fluctuations. It's something like the movie in the middle, that is holograms fluctuations. And when we combine many frames, we're able to increase the signal-to-noise ratio of these very tiny fluctuations and construct a power Doppler image of higher quality. So how do we do it in practice with subjects? Well, we have 3 cameras. There is one that is a pupil camera that is not very interesting to mention. It's just here to position the laser beam on the pupil. So we position the subject so that his pupil is facing the laser. Once that is the case, we ask the subject to fixate in the right direction. So most of the time we're interested in the optic nerve head region because that's where there are the most there is the more retinal vessels. When that is done, we make sure that the subject is not blinking and we press record on the ultra-fast camera. And after 1 hour of calculation, if we made everything right, then we obtain our high quality blood flow measurement. So in the eye fundus, there is 2 very interesting vascular networks. That is the retinal vasculature and the choroidal vasculature. The depth of field of the instrument is very large and it's able to include both of these layers, but in this presentation I'm only talking about retinal vessels. So this is the comparison between a commercialized instrument and our instrument. So on the left, this is scanning laser ophthalmoscopy, and the contrast of the image is only due to the reflectivity and absorption of the structures. So blood vessels appear in dark because the red cells have a higher absorption. They absorb the near-infrared light. And on our image of laser Doppler holography, there is a contrast of blood flow. So blood vessels appear with a bright contrast because where there is more blood flow, there is more power Doppler. So blood vessels appear brighter because there is greater local motion. So it's very nice to be able to have this blood flow contrast where you reveal the blood vessels non-invasively, but the real advantage of the method is really to be able to reveal the blood flow fluctuations over cardiac cycles. So all of the changes of blood flow during cardiac cycles, from systole to diastole, we can resolve them in a full field configuration. So in all vessels, in arteries and veins, we can measure these fluctuations and they bring some new information by themselves. Instead of imaging the average result of this blood flow fluctuation is what you would get with a slow instrument of very low temporal resolution, we can find some new information related to the biomechanical properties of the arterial system from these variations. So how do we make this movie? Because there is something that I've not mentioned, is that when you have a 2D imaging instrument with a very large depth of field, you get many Doppler contribution in the eye fundus because the eye is moving, so you have global eye movement. There is fixational lateral eye movement, but there's also axial movement from the pulsatility. You have capillary flow. Capillaries should be about everywhere, so they should create a Doppler signal. Then you have multiple scattering from choroidal blood flow. And what's interesting to us is blood flow in retinal vessels. So all of these contributions are in the images. And how can we separate them? So we're looking at three regions of interest here, retinal artery in red, retinal vein in blue, and the background in green. And what we see, if we take all of the Doppler frequency shift, that means a very low frequency threshold for the power Doppler, then we have something that is completely dominated by eye movement, because actually eye movements, they induce Doppler frequency shift at relatively low frequencies. So now let's take only the highest Doppler frequency shift, and what we see is something that has the pulse waveform that reveals it. It's related to pulsatility, but unfortunately it's the same in all regions of interest. And if we subtract the baseline signal is just a spatial average over the image, then we're able to reveal something that is specific to the feature that we're probing. So in the artery we see the pulse waveform, systole, diastole. In the vein we see something cycloidal, and in the background we see something flatter. This is what it looks like when we're making movies of it with and without the correction of the baseline signal. And the conclusion of this is that it's very important to make full field measurements to be able to measure the global contribution, to subtract it to the movie. If you're just making scanning measurements, then you cannot separate what is the global contribution and what is the contribution of the feature that you're probing. So that's why spatiotemporal filtering is necessary if you want to measure the proper waveform of blood flow. So in laser Doppler you can make several measurements on the Doppler spectrum. Here is semi-quantitative flow measurements, so it's semi-quantitative because it's still in arbitrary units, not in real physical units of flow or velocity. So power Doppler is supposed to reflect blood volume because, well, it's a quantity that reflects, let's say, the number of photons scattered by moving red cells, so it represents the number of red cells and thus blood volume. The mean frequency shift is supposed to reflect the mean velocity of the local flow, and we can make these two measurements. We measure something very similar, only it's very noisy with the blood velocity estimator. Here is a movie of the fluctuations of blood flow in an artery and a vein in a healthy subject. And what we see most of the time is indeed an arterial profile with a rapid increase of blood flow during systole, then decrease, a dicrotic notch, that is the small bouncing halfway through diastole, and then a slow decrease until diastole. And in the vein in blue we see something that is usually cycloidal. Alright, so what can we do with these flow variations measurements? So first immediate thing to do is differentiating arteries and veins. So if we take our power Doppler movie, we see that vessels have the same contrast on average because the average flow is the same in arteries and veins. But now if we consider the coefficient of variation calculated pixel-wise, so that is the standard deviation normalized by the mean. So where there are greater variations, as in arteries compared to vein, the coefficient of variation will have a greater value. So here we put a red to blue color map so that arteries come out in red and vein come out in blue. And that is a differentiation just based on the coefficient of variation. And this time you can make an image that distinguishes arteries from vein. Now more interesting is making resistivity maps. So retinal arterial flow, it is shaped by mechanical properties of the vasculature. So you have arterial compliance that plays a role, wave reflection and pressure amplification, and very importantly downstream resistance to flow. That is supposed to affect the waveform of the upstream arterial flow. So it's calculated as the value at systole minus diastole normalized by systole, that is resistivity index or the pulse law index, And with this image, you're also able to differentiate arteries and veins, and we measure something that is plausible in arteries, something about 0.7 for this subject. And this index is really important because it allows us to transform our semi-quantitative measurements in arbitrary units into an objective index that we can compare from one patient to another. So this time it's the variations of blood flow during cardiac cycle, the way it varies, that we can compare. And this should bring very important information about blood flow, how well the retina is oxygenated, as a higher resistivity index should mean a worse oxygenation. Now, in conclusion about this retinal blood flow presentation, this technology, laser Doppler holography, allows non-invasive, full-field blood flow measurements, and with a high temporal resolution thanks to the full-field feature. We're able to measure distinct waveform in retinal arteries and veins that seems to show that our blood flow measurements are very plausible. And this allows us to assess the resistivity index over the full field of view. Now, compared to the state-of-the-art instrument that I compared at the beginning of the presentation, the one that is most relevant to be compared to is laser speckle flowgraphy. And I think the nonlinear response to flow velocity due to the single exposure time of laser speckle contrast, we have something that responds better to a larger variety of flow velocities, and we're making more accurate flow measurements. And because of the coherent gain from the reference arm, we're able to have, I think, a higher sensitivity, which ultimately allows to detect better smaller vessels and mostly to have a higher temporal resolution. Okay, that's it for this presentation, and thank you very much for watching until the end.